Welcome to the Deep Dive, your shortcut to understanding the universe one fascinating topic at a time. Have you ever considered what fuels a firefly's flicker on a summer night? Or how a diver can launch herself off a high platform with such power?
Or even just, turning a page in a book.
Exactly. What connects all these seemingly disparate actions is energy. And today, we're taking a deep dive into the incredible, often invisible world of metabolism. We're pulling the essential insights from Campbell Biology in focus to give you a clear, concise picture of how every living thing, including you, masterfully manages its energy.
That's right. Our mission today is really to unpack the fundamental rules governing all of life's chemical reactions. We'll explore what metabolism truly is, the different forms energy takes, and the universal laws that dictate its transformations.
And then we'll get into how cells manage their energy budget, this amazing molecule called ATP, and finally, the role of enzymes. Those are the master speed regulators, right?
Absolutely. Think of them as the traffic controllers of the cell's chemical highways.
Hmm.
This deep dive should reveal some of the hidden logic behind what is essentially an incredibly complex chemical factory, the living cell.
Okay, let's unpack this. We should probably start by defining metabolism itself. It's often described as the totality of an organism's chemical reactions. But I think the key idea is that it's an emergent property. What does that really mean in this context?
It means the whole system metabolism has properties that you wouldn't necessarily predict just by looking at the individual chemical reactions in isolation. It's like an intricate roadmap. You have all these intersecting chemical pathways.
Pathways.
Yeah, metabolic pathways. A specific starting molecule, let's call it A, gets altered in a series of defined steps. A becomes B becomes Z, becomes D, which is the final product. And each step, critically, is catalyzed or sped up by a specific enzyme.
So enzyme 1 does A to B, enzyme 2 does B to C, and so on.
Precisely. And this is happening constantly. Sugars converting to amino acids, proteins being dismantled, all coordinated in this tiny microscopic space.
Right. So on this big metabolic map, you mentioned pathways. I gather there are two main types, like different directions of traffic.
You got it. We generally talk about catabolic pathways and anabolic pathways.
Mhm.
Catabolic pathways are the breakdown routes. They release energy by breaking down complex molecules into simpler ones.
Like demolition.
Kind of, yeah. The classic example is cellular respiration. Glucose, which is a complex sugar, gets broken down into carbon dioxide and water. And that process releases energy. It's a downhill reaction, energetically speaking. The cell captures that released energy to do work.
Like moving things across membranes or making muscles contract.
Exactly. All sorts of cellular work.
Okay, so if catabolism is downhill, breakdown, then anabolism must be the opposite, building things up.
Precisely. Anabolic pathways are biosynthetic. They consume energy to build complicated molecules from simpler precursors. Think about synthesizing an amino acid or assembling amino acids into a protein. These are the uphill reactions.
They require energy input.
They do. And the beauty of metabolism is how cells couple these pathways.
Mhm.
The energy released from the downhill catabolic reactions is often used to power the uphill anabolic ones. It's a very efficient energy economy.
This brings us right to the concept of energy itself. We throw the word around a lot, but in science, it's defined as the capacity to cause change, right? To do work.
That's the core idea. It allows things to rearrange, to move.
Mm,
And it exists in various forms. There's kinetic energy, the energy of motion. Think of water flowing or muscles moving.
okay.
Thermal energy, or heat, is actually a type of kinetic energy,
Huh.
the random movement of atoms and molecules. Then you have potential energy, which is stored energy. Energy stored due to location or structure.
Like water behind a dam.
Exactly. Or, crucially for biology, chemical energy. That's the potential energy stored in the chemical bonds of molecules, like glucose or fats. It's available for release when those chemical reactions happen, when bonds are rearranged.
The diver analogy helps here, doesn't it? Climbing the ladder. That's converting chemical energy from food into kinetic energy of muscle movement.
Which then becomes potential energy as she gets higher.
Right.
And then when she dives, that potential energy converts back to kinetic energy.
Which gets transferred to the water splash sound heat. The key takeaway is that energy is constantly being transformed, moving from one form to another.
Which leads us to the fundamental rules governing these transformations, the laws of thermodynamics. Now, scientists talk about a system, the thing being studied, like a cell or an organism, and the surroundings, which is everything else. And organisms are open systems, right?
Yes, that's critical. They exchange both energy and matter with their surroundings. They take in energy light or chemical energy from food, and they release waste products and heat. This constant exchange is vital for life. And the first law of thermodynamics, that's the famous one.
Conservation of energy. Energy can be transferred and transformed, but it cannot be created or destroyed. The total amount of energy in the universe is constant. A plant converts light energy. It doesn't create it. A bear transforms chemical energy from food. It doesn't make new energy.
Okay, so if energy isn't destroyed, why do organisms constantly need more? Why can't they just recycle it perfectly? That must be the second law.
That's exactly where the second law comes in. It states that every energy transfer or transformation increases the entropy, the disorder, or randomness of the universe.
Disorder. How does that work?
Well, during any energy conversion, some energy is inevitably lost as unusable heat, which is basically the random movement of molecules.
Hmm.
So when that bear runs, a lot of the chemical energy from its food is lost as heat to the surroundings. This increases the random motion of molecules in the air, increasing the overall disorder, the entropy.
So useful energy tends to dissipate into less useful, more dispersed forms like heat.
Right, and this helps us understand what a spontaneous process is in thermodynamics. It's not necessarily fast, it's just energetically favorable. It can happen without an input of energy because it increases the total entropy of the universe.
Yeah.
Water flowing downhill is spontaneous.
But pushing it uphill requires energy input. It's non-spontaneous.
Exactly.
Okay, but then, this seems like a paradox. Living things are incredibly ordered. Complex structures, organized cells. How does that fit with the universe tending towards disorder, towards higher entropy?
That's a really common point of confusion, but there's no conflict. Organisms create local order within themselves. They build complex molecules from simpler ones, arrange cells into tissues, and so on.
They decrease their own internal entropy.
Temporarily, yes. But they do this by taking in energy-rich matter and breaking it down, releasing heat and less ordered waste products, like CO2 and water, into their surroundings. So the total entropy, the organism plus its surroundings, always increases. Organisms are like islands of low entropy, maintained at the cost of increasing the entropy around them. Life doesn't violate the second law. It depends on this constant energy flow and entropy increase in the wider universe.
Okay, that makes sense. It's about the total picture. Now let's get more specific about the energy cells can actually use. You mentioned free energy.
Right, gives free energy, usually denoted as G. This is the portion of a system's energy that can actually perform work under the conditions inside a cell, constant temperature and pressure. We're usually most interested in the change in free energy, G, during a reaction.
Because that tells us if the reaction had happened on its own.
Exactly. If urg is negative, the reaction releases free energy. The system becomes more stable, and the reaction is spontaneous. It can be harnessed to do work. If urg is positive, the reaction requires energy input. And what happens when Ag is 0?
That means the system is at equilibrium. It's reached maximum stability, its lowest free energy state for those conditions. And crucially, a system at equilibrium cannot do work. This is why a living cell can never be at equilibrium.
If a cell reached equilibrium, it would be
Dead. Life is a constant process of avoiding equilibrium.
So, based on this EG, we categorize reactions as exergonic or endergonic. Exergonic sounds like energy exit.
That's a good way to think of it. Exergonic reactions have a negative G. They release free energy, they're spontaneous. Cellular respiration is a prime example. Breaking down glucose releases a lot of free energy. It's energetically downhill.
And endergonic is the opposite, energy inward.
Right. Endergonic reactions have a positive G. They absorb free energy from the surroundings. They are non-spontaneous. Synthesizing complex molecules like proteins or DNA requires energy input. It's energetically uphill.
So how do cells manage to run all these necessary uphill endergonic reactions? They use a strategy called energy coupling. They basically use the energy released from an exergonic reaction to drive an endergonic one. And the main molecule involved in this coupling, the cell's primary energy shuttle, is ATP.
Adenosine triphosphate. Okay, tell us about ATP. Cells need it for, well, pretty much everything, right? Chemical reactions, moving things, contracting muscles.
All of that. We categorize it as chemical work, like synthesis, transport work, pumping substances across membranes, and mechanical work, movement. ATP powers all three.
What does it look like, and how does it store energy?
ATP consists of an adenine base, a ribose sugar together called adenosine, plus a chain of 3 phosphate groups. The key is the bonds between these phosphate groups. When the terminal phosphate group is broken off by adding water, a process called hydrolysis, ATP becomes ADP, adenosine diphosphate, plus an inorganic phosphate molecule, pi.
And that releases energy. People often talk about high-energy phosphate bonds. Is that accurate?
It's a bit misleading. The energy isn't stored in the bond itself, like a packet. Rather, the reactants, ATP and water, have higher free energy than the products, ADP and pi. The release of energy comes from the chemical change to a state of lower free energy. Think of it like a compressed spring. The energy is released when the spring uncoils and reaches a more stable state, not stored within the coils.
Okay, so the instability of that triphosphate tail is key. How does this hydrolysis actually power work?
Usually through phosphorylation. ATP transfers that terminal phosphate group to another molecule. This phosphorylated molecule, called a phosphorylated intermediate, is now less stable, more reactive.
So it primes the molecule for whatever reaction needs to happen.
Exactly. For chemical work, this makes an otherwise endergonic reaction proceed because the overall coupled process becomes exergonic. For transport and mechanical work, ATP binding in hydrolysis often causes a protein to change its shape, which results in action like a transport protein opening or closing, or a motor protein taking a step.
It's amazing how versatile it is. But cells use a lot of ATP. They must remake it constantly.
Oh, constantly. That's the ATP cycle. ATP is hydrolyzed to ADP and pi, releasing energy for cellular work. Then, energy released from catabolic exergonic reactions, like cellular respiration, is used to reattach the phosphate group to ADP, regenerating ATP. This regeneration is endergonic.
So it's a constant cycle.
Yeah,
Break down ATP to get energy. Use energy from food to build it back up.
precisely. It shuttles energy.
Hmm.
And the rate is incredible. A working muscle cell might recycle its entire pool of ATP every minute. We're
Right.
talking about something like 10 million molecules of ATP consumed and regenerated per second in a single cell.
Wow. That's staggering if that regeneration didn't happen.
We'd use up our body weight in ATP incredibly quickly. The cycle is absolutely essential.
Okay, so we have energy, we have ATP as the currency. But you mentioned earlier that even spontaneous exergonic reactions can be very slow on their own, like sugar just sitting there.
Exactly. That's where enzymes come in. They are biological catalysts, mostly proteins that speed up metabolic reactions without being consumed by the reaction. Without enzymes, metabolism would essentially grind to a halt.
Why are reactions slow without them? What barrier are they overcoming?
It's called the activation energy barrier, E. Before a reaction can occur, the reactant molecules usually need to be contorted into an unstable, high-energy state called a transition state. Think of it like needing to push a boulder slightly uphill before it can roll all the way down the hill. That initial uphill push is the activation energy.
And enzymes somehow lower that initial hill.
Precisely. Enzymes work by lowering the EA barrier. They make it easier for the reactants to reach the transition state. Importantly, they don't change the overall free energy change of the reaction. They can't make an impossible reaction possible. They just make a possible reaction happen much, much faster.
And they're very specific, aren't they? One enzyme for one reaction.
Highly specific. The reactant an enzyme acts on is called its substrate. The enzyme binds to its substrate, forming an enzyme-substrate complex. This specificity comes from the enzyme's unique 3D shape, particularly a region called the active site.
The active site is where the magic happens.
That's where catalysis occurs, yes. It's often a pocket or groove on the enzyme surface. And it's not usually a rigid fit like a lock and key. More often, it's an induced fit. When the substrate binds, the active site changes shape slightly to bind the substrate more snugly, kind of like a handshake tightening.
Clever. So what exactly does the enzyme do in the active site to lower that activation energy?
It uses several mechanisms. It might orient the substrates correctly relative to each other. It might stretch or bend the substrate's chemical bonds, making them easier to break. It could provide a favorable microenvironment, like altering the pH locally. Or sometimes the enzyme even participates directly in the reaction by briefly forming covalent bonds with the substrate.
And they work incredibly fast, too.
Astonishingly fast. A single enzyme molecule can process thousands of substrate molecules per second.
Are enzymes affected by things like temperature or pH?
Very much so. Each enzyme has an optimal temperature and an optimal pH where it works best. For human enzymes, optimal temperature is usually around body temp, 37 degrees C, much higher, and the enzyme can denature, unravel, and lose its shape and function. Optimal pH also varies. Stomach enzymes like acidic conditions, intestinal enzymes like slightly alkaline ones.
Do they ever need help, like non-protein helpers?
Yes, many do. These are called cofactors. They can be inorganic metal ions like zinc or iron, or organic molecules called coenzymes. Many vitamins are precursors for coenzymes, which is why vitamins are essential nutrients.
Okay, so enzymes speed things up, but can things slow them down? Are there enzyme inhibitors?
Absolutely. Enzyme inhibitors are molecules that interfere with enzyme activity. Some are competitive inhibitors. They resemble the substrate and compete for binding to the active site. Others are non-competitive inhibitors. They bind to a different part of the enzyme, causing it to change shape so the active site works less effectively.
Are inhibitors always bad, like poisons?
Not at all. While some poisons and drugs work by inhibiting specific enzymes, many natural molecules within the cell act as inhibitors. This inhibition is actually a crucial way the cell regulates its own metabolism.
Right, because you wouldn't want all pathways running full blast all the time. That would be chaos.
Exactly. Chemical chaos.
Uh-huh.
Cells need tight control. One major way they achieve this, besides controlling enzyme production, is by regulating the activity of enzymes that are already present.
How do they do that?
A key mechanism is allosteric regulation. This happens when a regulatory molecule binds to the enzyme at a site other than the active site called an allosteric site. This binding changes the enzyme's shape, affecting the active site's function. It can either activate or inhibit the enzyme.
So a molecule binding somewhere else controls the main business end.
Precisely. Many allosteric enzymes are complex, made of multiple subunits. The binding of an activator molecule stabilizes the active form of the enzyme, while an inhibitor stabilizes the inactive form. It's like a molecular switch. ATP and ADP often act as allosteric regulators for enzymes involved in energy metabolism, providing feedback on the cell's energy status.
And there's also feedback inhibition, which sounds like a loop.
It is. It's a very common control mechanism where the end product of a metabolic pathway binds to and inhibits an enzyme that acts early in that same pathway.
So if the cell makes enough of product D goes back and shuts down enzyme 1 that started the A to B conversion.
Exactly. It prevents the cell from wasting resources by making more of something it already has enough of. It's a self-regulating system. Very elegant.
And finally, these enzymes aren't just floating around randomly in the cell soup, are they? They're structure.
Definitely. Cellular structure brings order to metabolism. Sometimes, enzymes that work together in a pathway are grouped into multi-enzyme complexes. The product of one enzyme is passed directly to the next, increasing efficiency. Enzymes can also be embedded in specific membranes or localized within specific organelles, like mitochondria, creating specialized compartments for different metabolic processes.
Wow, it really is an incredibly intricate, organized, and efficient chemical factory.
It truly is.
Okay, so let's recap. We've journeyed through the world of metabolism, the sum total of life's chemical reactions. We saw how it's governed by the laws of energy, particularly the drive towards increasing entropy, and how cells manage their energy using the currency of ATP, constantly cycling it.
And we saw how enzymes act as essential catalysts, lowering activation energy barriers to make reactions happen at life-sustaining speeds, and how their activity is exquisitely regulated through things like allosteric control and feedback inhibition, often organized within specific cellular structures.
It gives you a much clearer picture of the sheer dynamism inside every living cell. And this isn't just abstract biology, is it? Understanding metabolism helps us understand health, disease, how medicines work, even ecology.
Absolutely. It's fundamental to basically all aspects of biology and medicine. How our bodies generate heat, how plants convert sunlight, the mechanisms behind metabolic disorders like diabetes, even how cancer cells fuel their rapid growth. It all comes back to these core metabolic principles.
So, as a final thought for you, our listeners, consider the sheer scale and speed. Millions of ATP molecules turning over every second in every cell. Enzymes performing thousands of catalytic cycles per second. This incredibly complex, tightly regulated dance of molecules. What does this constant, efficient chemical transformation tell us about the nature of life itself? About its resilience, its adaptability, its inherent elegance? What new questions does it spark for you?
Thank you for joining us on this deep dive into the fascinating world of metabolism. We hope you feel a bit more informed and maybe even a little awestruck by the chemical marvels happening within you right now.